Okay, so one of the questions I got asked a lot on the first two parts of investigation 4 was, do I have to do 3 and 4? Yeah, I think, I'm pretty sure I had said it in class, just do 1 and 2. 3 and 4 are for anyone that's looking to go above and beyond. There's no extra credit or anything, but if you're looking for additional challenges to help solidify your understanding of what we're doing here for the membrane dynamics portion of this unit, then by all means do that. Those were some of the experiments that I would have done had we been able to do some labs and as we've done in previous years, but it's not required. And there's a video there that talks about some of the types of examples that you might see on the test. I think it was number 4. So if you wanted a little more work to prepare yourself for the test, that resource is there for you. There's a video on YouTube, and you can use that time to practice making these artificial cells. The more practice you do, the better off, more comfortable you'll be for the test. That's all. As long as you've shared with me that you did 1 and 2 and you practice graphing and you practice thinking about these artificial cells in that second scenario, right, where you're putting them not in water, but in 0.4 molar sucrose, you're good. Okay, so as long as you did that graph and answered those questions, you're in a good spot. I'll give you some lab points for that. The second thing that we want to do for this investigation, the one that I would have really loved to have done with you, is this osmosis in living cells. So we choose various types of tubers, right? And so each of these is different in their polysaccharide content, depending on the plant that produced them. So this would have been a really fun lab to do, but again, it's not going to work out. So I will guide you all in this room to, and at home, I'll share this with you. I haven't shared it already, to do this, okay? So go through and read this scenario. It's very similar to the previous lab challenges, except now we're working with actual tissue from a living thing instead of an artificial cell, okay? Same idea though. You're gonna encounter these mystery solutions, okay? So of these mystery solutions, so they're colored and labeled A through F, one of them is 0, one of them is 0.2, one of them is 0.4, one of them is 0.6, one of them is 0.8, and one of them is 1 molar sucrose. You don't know which is which, okay? Your job in this lab challenge is to tell me which of those you think is what solution, okay? So in the end, you're gonna say, I think A is water because of this, right? Here's the reasoning. Here's my data to support what I'm saying. You'll graph out your data. All of that is outlined here. I'll walk through this with you real quick. Okay, and then there's this page here to click on that actually has the questions and lab report that I want you to write, okay? Our next class is a free space for us to catch up and review the first half of this unit, okay? So we're not moving on to metabolism just yet. There'll be one more class for Q&A, for work. So this won't be due until after that class. That doesn't mean you should do math, you know, when I cut you loose here in a few minutes. That means that you have some more time to do these things and to get these concepts right in your mind before we move on, okay? So going back through this, I would like to share with you what this procedure looks like really quick. It's a fun one. Not a boring next one. Okay, you guys at home, hang tight for 1 second. So, had we been in groups, what you would do is you'd take your tuber of your choice. So you have like, I don't know, russet or a sweet potato, and you would take these cork boards and drive them into your potato. So it's really fun and cathartic to stab your potato and produce a little core sample. Okay? So this is your experimental subject, this tube of potato cells. Okay? Now I would take this and I'd cut off the skin. And I would try to produce samples that are about that size. Okay? So I would say normally I would ask for probably 3 of these per sample. Okay? So I keep them in the weigh boat. I cut off the skin and then I continue to cut out my core samples. Now you want to be kind of careful and economical with your potato. Why is that? Why do you want to have all of your samples ideally from the same potato? These are sort of fun things that I always ask about because in science, the challenge is to design an experiment to answer the questions that you're asking. So that means procedurally you have to do certain things a certain way to do that. So that's the fun problem solving part of science. So this potato is a unique potato, right? Whatever happened in the plant to produce this, these things are going to probably be similar to one another. Whereas if I get another potato, it's like, is that going to be like the potato's brother? It is. I can say this potato and this other potato, they're closely related. No, like whatever happened in that potato is going to be different than what happened in this potato. So ideally, you want to be economical to the point where you're getting all of your samples from the same potato. Not always possible, but that's the goal. So then I put them in the weigh boat. And for example, this, if I had imagine there's 3 in here, I would put these 3 samples on the scale. I would weigh them as an initial mass. Then I would put them into one of the colored solutions overnight. Come back in the morning and do my little blotting thing. Imagine I'm taking these out of the solution. I put them onto that paper, and then I do this. Why is that? To blot off the excess solution, and then they go onto the scale again, and that's their final mass. Okay? So you'll see a map that I've given you the data. It's now the next day. You don't have to collect your samples, but you do have to calculate the percent mass change. Okay? So that's where we're at with this experiment for you guys. Any questions? Okay, so I'll cut you loose on this assignment. I'm going to talk to our online friends. So you're free to do your work. Okay, I'm going to work with these guys for a minute. But, be productive. You guys still there?
Yes.
So on the assignment sheet, and let me make sure that I have this posted for everyone. I'll put it on the bulletin board, okay? Okay, so that link should be there on the bulletin board for you, and that goes for you guys at home too. But really quickly, you couldn't see what I was doing before. this is your research subject today. Jerry, what kind of potato is this?
I have no clue.
You didn't study your potatoes last night in preparation for the lab?
I know if you actually wrote an essay about potato freshman year.
That's right. Well, this is an Idaho russet, okay? So we're gonna be taking core samples from this. To do that, I use this cork borer. So I put the cork borer into the potato, and I press through it. Hang on, I want to change the view of this. Okay, so I do that. I take that out. I want to pop my sample out. So here's your sample for the lab. It's this potato tube. Okay. I'm going to put it into a weigh boat. It's plastic. Trim off the edges. So I want these things to be roughly the same size. And I take these 3 potato cores to the scale in my weigh boat, and I weigh them for their initial mass. Okay. Those potatoes would then go into one of the unknown sucrose solutions overnight and would normally be masked the next day. First, I would do this. Why would I put them into the paper towel like this and kind of blot really quickly after coming out of the solution? Because there
I guess
would be some external solution that could increase the mass artificially, right? So I blot it to remove that confounding factor. And then I put them on the scale to weigh them for a final mass. So at that point, you could calculate the percent mass change. So you'll pick the experiment up here online by looking at the data table that I've given you, showing initial and final masses for samples. Okay, then just complete the other components of the lab report. Fair enough? Any questions?
good.
Okay. If you guys don't have any questions, you can go and work on that asynchronously. Okay?
Mm-hmm.
See you next.